Welcome everyone to this month's webinar. This time, like summer, a lot of vacation time. So we also have kind of a partly vacation of the webinar with only having one talk today. Just reminders, some reminders at the beginning. Again, there are still slots for the winter and fall session for trainee talks. We're pretty filled, luckily, with senior scientist talks. But if you're students, or if you are a student, you're interested in participating at the webinar, and if you want to contribute and distribute your work here, you're always welcome to just drop us an email. We're happy to include you at this webinar. Another reminder is the YouTube videos. We are recording these webinars, and most of them are actually already published on YouTube. So will also be this webinar today. Marcin just agreed that it can be published on YouTube. Thanks for that. So just if you are interested in the previous talks, also including Bruno's talk, for example, you can just type in EuchisWebinar at YouTube, and you will find all the videos and the webinars, the past webinars. With that, I would like to introduce you, Marcin. Marcin is a graduate of the Technical University of Łódź, Poland, and he obtained a PhD in organic chemistry in 2001 at the University of Potsdam. And I just learned that Martin Peter, right, was your supervisor there.
Right.
Yeah. You followed up with a habilitation in Łódź in 2008. And in 2008, Marcin was also started as the director for science at the Institute of Security Technology, MORATEX in Łódź. And with 2016, right? Since then you have been acting as the director of MORATEX, and I would call it during your regency. There were 4 medical devices implemented for commercialization, including wound dressings and applicators for implants and so on. And you have further medical devices in clinical and preclinical studies. And beside that, and I guess you will give us an introduction about MORATEX. Your work there also resulted in 5 technologies of ballistic protectors, and also 2 textile solutions for fire guard protection. And clearly, chitin and chitosan has been playing a pivotal role in your R&D activities. And Marcin, really curious to learn about how a security technology institute and chitosan goes together. So happy to listen to your talk today. Please go ahead. The floor is yours.
Okay, thank you very much, Gregor. Good morning, everyone. Good morning in the virtual society. I have the great pleasure, ladies and gentlemen, to present you shortly our experiments in a case of designing of advanced medical devices, mostly hemostatic topical agents, for application of the special area of use, that's mean security and defense area. Before starting the main subject of my presentation, I would like to introduce you Institute of Security Technology. We talked before the presentation with Gregor about, however, the MORATEX is a very small institution because we have all employments is around 70 people, and all scope of interest is very wide because we are responsible for the designing several types of product equipments and uniforms, which is usable in the security area. We have also 3 accredited laboratories for meteorological, chemical, and ballistic study, and we are also a notified body for the certification of personal protective equipment. And our supervisor is Minister of Interior and Internal Affairs and Administration, which indicate that all our activities focus on the services which is supervised by the minister, that's mean police and border guards. And on this picture, as you can see, some some examples of our product which we designed during our last 20 years. As I told, we are responsible for the designing of the protection vest. Mostly there are some ballistic vests, heat protection, ballistic shields, and anti-blow product. However, we also, our interest is also focused on some medical devices which is possible to use in security area. Because as you see on this citation, the several data confirms need to develop some modern hemostatic topical agents due to uncontrolled hemorrhage accounted up to 50% of the total death. In other cases, in the case of active war conflicts, for example, some conflicts which was carried out in Afghanistan and Iraq, 19% of combat participants died before the surgical supports, and approximately 90% of potentially survivable deaths resulting from hemorrhage. In the case of civilian hospitals, because some technology which we usually call the dual-use technologies is possible to adopt in the civil area. In the case of civilian hospital practice, traumatic injury is the second case of death, amounting to 10% of deaths, in which up to 40% mortality due to hemorrhage. That was a need which we start to know about the possibility to design the new solution for activate the hemostasis in the case of hemorrhage resulting from the typical action of the services acting in the security area. And the first work research we started in 2005 with a cooperation with Tricomet. There's a Polish company which produce implantable medical devices, and also our partners, Institute of Biopolymers Chemical Fibers. And this work was finished in 2013 by the CA certification and the implementation to the practice of the first hemostatic wood dressings. I will tell later about the characteristic of this medical device. Parallelly, since 2010, we started with a new project aimed to the selection of the materials and the system of the materials for the maximization of the performance, including hemostasis. And the result of this project was, the output of this project was other projects. The third project was started in 2013, which aimed to prepare several proposals of topical hemostatic topical agents, which is possible to use for the completion of medical kit applicable in the security area. Now it's 2021, and we are doing the clinical trials. And the first adventure for the application of chitosan for designing of medical devices was ThromboGuard. As I told, this product, which was these materials we tried to develop in starting in 2005, and the aim of our activity was to design a new generation of hemostatic wood dressing for application in special area of use, as well as we tried to adopt this solution for the civilian area, that's mean for the typical hospital. On these pictures, you can see the scheme of idea of a ThromboGuard, which is made of 3 layers. outer layers is made from the polyurethane films, which select, pass gases and protection against external factors. And this outer layer is responsible for the activation of exogenic blood clotting by the direct pressing of in trauma. And the middle layer, made of a hydrophilic polyurethane foam, which very innovative structure, so-called pore-in-pore, which have a very high absorption affinity, is responsible to the absorption of high volume on morphotic blood constituent that result in the increase of the concentration of clotting factors in place of trauma. And the main active layers is a last layer of containing the active factors initiating the exogenic pathway of the blood clotting, made of the chitosan, alginate, sodium and calcium alginate. With there is also some option which with this layer contain also some silver particles for the exchange antibacterial properties. And this layer is responsible for the active polarization of affective factors initiated an exogenic blood clotting pathway. For example, chitosan activate platelets aggregation due to the positive charge on the polymer chain, and the calcium activates the plasmatic clotting agents. During our clinical studies, we started to determine the several factors which will characterize the properties, performance of the ThromboGuard, and we typed two types of factors. This means moisture vapor transmission rate, which measures the breathability of the wood dressing materials, and also fluid handling capacity. As you see, these parameters was very high in the case of ThromboGuard, which was supporting us for decision to use this type, this scheme, this system of layers which we designed. We also check, unfortunately, there should be some percent, not this data, forgive me. We also check the biological behavior of these materials in two types of tests against the Staphylococcus aureus and Escherichia coli to check the bacteriostatic activity and antiseptic activity. And for this system which we developed, we obtained very high bacteriostatic activity and as well as very high antiseptic activity. We also prepared the acceleration aging study to check how this bacteria bioactivity will change during the time. In the case of antiseptic activity, we obtain some changes, some insignificant changes in the bacteriostaticity from 3 to 11% after the 1 years of accelerated study, whereas in the case of bacterial stacity, the changes was 0 or 0 or near to 1%. During the research, we carried out several verifications in a range of performance and the safety using tests in vitro and in vivo. And the ThromboGuard safety was confirmed in the cytotoxicity, irritation, subacute reactivity, allergenicity, hemolysis, and hemostasis, as well as wound healing. We tested these prototypes of ThromboGuard on the animals. And we also test the pyrogenicity because there is a presence of chitosan which can be also pyrogenic. After the preclinical study, we decide to start the clinical study. the aim of this clinical study was double. Primary, there was estimation of safety and performance, effectiveness of hemostatic and antibacterial on behalf of a ThromboGuard for short time of use, maximal for 5 days, and for long term, for verification of application place after 1 to 3 months, due to confirm the safety of a new composition which we implemented on to the clinical practice. And the secondary aim was to compare the effectiveness of the action of the ThromboGuard against other reference wood dressing. In this time, there was a 2010, there was no on the market, there was no this equivalent of the ThromboGuard when we compound from one wood dressing. It was consistent for Surgicel, which is characterized by the hemostaticity, and the Medisol silver pad, which contains silver for the antibacterial properties. And there's a combination of these two wood dressing was a reference during our clinical study. During the clinical study, we compare with the referenced wood dressing the direct hemostatic effect stability during first 15 minutes after 24 hours, 2 days and 5 days, And also we compare the comfort of use, some aspect of possibility to remove, and the other clinical advantages. And last comparison which we took, there was antibacterial effectiveness based on the testing of inoculation from the wound of the patient. On this picture, you can see the effectiveness of hemostasis for the ThromboGuard and the controlled wood dressing. And we observed a significant reduction in bleeding time, and that also some control possible to control by the ThromboGuard of bleeding from the wound. And this hemostatic effect was stable during first 24 hours. We also confirmed that wood is prevent against infection for 24 hours. After 24 hours, we also control the reduction in bleeding, as well as the stability of hemostatic effects, as well as prevention of wound infection for this until this time. And we obtained the high efficiency of the ThromboGuard in comparison with the control wood dressing in the case of hemostasis, as well as prevention of wound infection. On the slides, you can see the ThromboGuard, which is now on the market because we certified this wood dressing in 2012. In 2013, it was implemented on the market. And the clinical study, which we performed in 2010 to 2012, showed a high hemostatic effectiveness. That means a repeatability for 100% of the patient. That means some whole population of the patients, and a very quick hemostasis till 3 minutes, and high antibacterial effectiveness, because we confirmed the protection of wound against infection till 5 days. Clinical universality, and what is most important in case of patient, that means some safety. That means absence of allergenicity, adverse effects connecting to the hematology, biochemistry, and microbiology. And the second part of our experiments in the case of designing of hemostatic topical agents was works, which we started in 2010, which aimed at screening the materials and some system for design of new generation of hemostatic topical agent system for potential application in special area of use. We selected two types of technology which we used for the production of the formulation of some composition of hemostatic agents. First, there was a freeze drying, which produced a particle in size ranging from 0.9 to 6 micrometers. And we used two types of formulation. That means first there was a chitosan alginate in the form of sodium or calcium salt, as well as this formulation was additionally fulfilled by the carboxymethyl cellulose. Second technology which we used was spray drying, which resulted in the powder in particle size ranging 10 to 60 micrometers. As reference, we used two types of hemostatic wood dressing. There was a Celox wood dressing, which is characterized by the particle size of powder 0.1 to 0.9 millimeters, and a quick clot wood dressing in particle size 1.8 to 2 millimeters. We also compared these materials in the case of moisture vapor transmission rate, as well as absorption under the free soaking. And we obtained that all materials are characterized by very high, approximately 2 times higher moisture vapor transmission rate as compared with Celox, and 2 times lower absorption under tree soaking, as we also compared with Celox. We also compared these materials with reference again to check the bacteriostatic activity and some bactericidal activity, and we found that some composition which we obtained from spray drying have very high bioactivity in the case of bacteriostatic and bactericidal activity, comparable to the Celox. However, for both or a composition made from the chitosan and sodium or calcium alginate, the bioactivity was much more high as compared with the clicklot. We also checked several properties. We verified several properties which are responsible for the safety of the designing medical device in scope of cytotoxicity, irritation, C-reactive reactivity, some allergenicity, and we performed also some very wide scope of compatibility test, especially in the case of coagulation affinity and recalcification affinity. And those tests allowed us to select the most optimal formulation for future works to design a new generation of hemostatic topical agents. And the last part of my presentation will be some aspect of designing of medical kits, which consist in several usable form of hemostatic topical agents, which we select, as I told before. And the aim of this activity was to design a new idea of medical kit, as I told, containing various usable forms of hemostatic topical agents, especially for the application in the security area. Again, we test the physical properties of this parameters in the case of moisture transmission rate as well as absorption under free soaking. However, we use the usable form. There was two types of usable form of composition of chitosan and sodium or calcium alginate. First, there was lyophilized foam, and the second, there was impregnated ceton. As you see, all this usable form are characterized in the case, especially in the case of lyophilized foam, showed the similar properties as compared with the Celox, especially in the case of absorption under free soaking. However, in the case of moisture vapor transmission rate, all designed usable form of topical agent have very high moisture vapor transmission rate, approximately 2 times higher as found in the case of Celox. We also prepare some test of accelerated aging, because we have to know how the performance of a usable form of topical agents will be changed during the time of storing. As you can see on these figures, the changing in the case of absorption free, under free soaking or MVT-R are very low. Approximately 10% lower. We detect approximately 9% lower MVT-R after 1 year of accelerated aging. And in a case of absorption under free soaking, the changes was not higher than 4%. On these pictures, you can see microphotographs and microphotographs. You can see the structure of first usable form of hemostatic topical agent. There is a ceton with which we prepare from the woven gas impregnated with the formulation of chitosan with sodium and calcium alginate before accelerated study and after accelerated study. This changes was not so insignificant in the case of view of these materials, and after this 1 year of accelerated study, we also did not found any changes in the activity, bioactivity of these materials. However, in the case of foams, lyophilized foams, after 1 year of accelerated study, we found the quite different structure of the surface, because probably there is a connected with the migration of glycerin, because we use glycerin as a plasticizer of this composition. These particles will, during the accelerated aging, will transfer to the surface of foam. And there is probably last slide. There on the slide you can see the materials which we transferred to the clinical study. There was three types of usable forms of hemostatic topical agents, which is a part of medical kits. First, there is a hemostatic wood dressings made from using the freeze drying in the form of foam. Second, there is a tactical gas, which is designed from the impregnated ceton. And unfortunately, for the third, I cannot give you any information because it's very confidential in the case of mode of action of this medical device. There is a hemostatic compression bandage. The confidentiality is especially in the case of method of the compression, which is very easy in the case of this type of wood dressings. And the last slide, our experiments in the case of application of chitosan for designing of medical devices was started in 2005. In 2013, that means after the 8 years, we implemented on the market the first product. The range of conformity which is necessary to fulfill is very wide, especially in a case of confirmation that chitosan is safe in a case of potentiality to transfer any pathogen from the animals because there is animal tissue derived substance. In 2010 to 2013, we prepared several tests for the selections, optimal hemostatic composition and formulation as outputs for design a new generation of a hemostatic topical agent, which we use in next step or experiments in the case of chitosan use. During last project, which was started in 2013, and during this project, we tried to develop a new ideas of different types of hemostatic topical agents. And now this hemostatic topical agents are doing the clinical study. I hope this clinical study will be finished this year. However, we have a COVID pandemic, and the clinical study was stopped in 2020. Now we started again. And I hope in next years this medical kit will be on the market. And the last, because I have also last slide, is very important, because during our experiments in a chitosan application, we have only one conclusion, that as you can see on this picture, there's pictures of composition of Banksy, which told the key to the making of great art is all in the composition. It's true. It's also true in the case of research, in the case of testing, and the making of great science and great product is all in the composition. Whereas not in some composition, some idea, some thinking is not applicable in the project, the product which we develop will be not functional. There's also very important in the case of safety of these materials, because these materials should be safe for the patient, and that's it's very important. I told again, it's very important to have the knowledge, the experiments, and also to design the project track to obtain a safe and functional product. Thank you very much.
Thank you so much, Marcin, for this interesting presentation. Really curious. I was really curious and interesting to see all the products out there on the market of what you're developing. Are there any questions out there so far to Marcin?
May I have a question?
Martin, please go ahead. Do you have a question?
Yeah. Okay. Marcin, do you have any European or international patents? This is one question. And the second question is, what is the regulatory status? You mentioned that you hope it will be in the market next year. Are you through with the regulation, or what is the status right now?
Okay, first question. Unfortunately, we haven't got any patents, European patents and international patents. We have the regional Polish patents for the ThromboGuard. In the case of composition, which we developed during the second part of our studies, we have 4 patents, also Polish patents. However, in the case of last project, there is some problem in the case of possible to publish the information, because all this result will be will be applicable in defense and security area. That until now we have not any confirmation from Minister of Defense and Minister of Interior to have possibility to protect this idea in some Polish or European regulation.
Mm,
The second question, we have now we are doing the assessment of the essential requirement for new regulation of European Union, because probably you know that 2 years ago the regulation was changed. We have now assessment of requirement for the MDR. There's a new regulation of European Union in case of medical devices. Mm-hmm. And I hope there will be no problem, because in the case of implementation, because after the finish of the clinical study, we are starting with the CA certification by notified body. Fortunately, because I told that now we have the problems with the clinical studies, because between 20 to 21, this clinical study was stopped due to COVID pandemic. Yeah. Fortunately, this time was very good for us, because the companies, that means notified body, reduce maximally the payment for the CA certification. When we firstly, when we tried, according to the plan of the project, we should to start with CA certification on the beginning of this year. 6 months late, we have a quite different cost. That means the reduction cost of confirmation by CA mark. That means I hope into next year these medical kits will be on the market.
Thank you very much. It looks very interesting, and there should be no problem, hopefully. Thank you. Bye-bye.
Thank you.
Okay, next question is from Inma.
Hi, thanks very much for the talk. I'm going to remove my mask. Wait for a moment. Are you hearing me clearly?
Yeah.
Okay. I have a question and a curiosity. The question is that I have seen at the beginning of your talk that you were talking about the use of silver nanoparticles as an optional possibility. But in the rest of the talk, you have don't talk about this possibility. I don't know if it is because you decided not to continue with this line. And the curiosity is about the clinical test, because I have seen that it looks like the patients were without hair in the legs. So it's because it's part of the assay, or can you comment on this? Because I'm not an expert on this field, so I don't really know why they remove the hair of the legs.
Okay, first question. During our first study, we use particles of silver to exchange the action of antibacterial action. However, there was a problem with the certification, because you probably know that this material, which containing some drug, and silver is a drug, is a so-called medical device borderline product, because there is medical device containing drug. And the cost of this confirmation of safety and performance was very high. So that was the first decision. And the second information, there was absence in Poland, some regulation for the registration of a medical device which contain drug. That after this, we decide that it was necessary to remove silver from the thrombogard and use only some active layers containing chitosan. As you saw the result of bactericidal and bacteriostatic properties, only chitosan have very high bioactivity against the bacteria, that we decide to remove silver from our prototype. And then we did not use any drugs because the application of drug will increase the cost of the project. That is the first. And the second, prolongate the term of CA certification, registration, implementation on the market. But activity of chitosan in case of antibacterial action is very high, and we confirm during the validated clinical study. And then I came to the second question. There is a problem too in a case of clinical study for simulate the real condition of use of hemostatic topical agents. Because as you probably know, the clinical test needs the patient participation in the decision to go to the clinical test or not. That we decide that for clinical tests we will use some person after the transplantation of the skin. This clinical test was conducted in the Military Institute of Medicine in Warsaw, and we hope this data are very validated, and also it was confirmed by two regulation office, that means the Polish registration agency as well as notified body.
Okay, thank you very much. Very nice.
Interesting, yeah. Are there any more questions from the audience? If not, maybe I ask one question out of personal curiosity, maybe the curiosity from Euchis side. What was the source of chitosan that you used? Can you comment on that? And maybe
the source, yes, I can. The source of the chitosan was chitin from the shrimps. As I told you, we usually use two manufacturers, so two chitin from the two manufacturers. That's mean Hepe from Germany, and from Iceland. The future last area of name was Vanson, probably.
Okay, I see there's a question in the chat too. I think that was answered right away. And adding on to the questions I asked, the question in the chat also asked for the molecular weight and the degree of deacetylation of the chitosan used. Is that, are you aware of that? I guess if you buy it from
yes, yeah, I can answer for this question. We use chitosan with molecular weight between 2,000 kilodaltons to 3,000 kilodaltons. 2,000 kilodaltons to 300 kilodaltons. About the acetylation degree is between 80 to 90%.
Okay, thank you. Well, I'm curious about also the consistency of the raw material. Did you observe, like in your manufacturing process, when you make these foams, for example, for the ThromboGuard, did you experience any like starting material inconsistencies that all of a sudden the foam formation was different or anything? Or is the consistency of the like starting material, meaning chitosan in that case, was that always like perfectly consistent and you never faced any issues there?
We never observed this phenomena. Okay. There's probably, as I showed, that one phenomena it's problematic to understand. There is probably some migration of glycerin plasticizer to the surface of foams after the accelerated study. However, accelerated study is conducted in the high temperature. We usually use temperature between 55 to 60 centigrade Celsius. That means probably this temperature also affected the migration of the glycerin to the surface of the foam. Because we did not observe this phenomena in the typical accelerated, that means accelerated at real condition samples.
Okay, very interesting. Okay, last chance to ask any questions before we close the session for today. I don't see any raised hands or any comments in the chat. So thank you again, Marcin. Really interesting talk. Really interesting to see the application side of chitosan. And yeah, thanks so much. Thanks everyone for participating at today's webinar. We'll most likely not have a webinar next month. It depends. It's the start of the semester again. Everyone seems to be really busy at that time. So maybe our next seminar, but not finally determined, is the end of October with Katja Richter from Medical chitosan. So with that, everyone, have a good day and see you next or in 2 months.
Thank you very much. Bye.
Everyone.
Thank you very much.
Thank you, Gregor. Thank you, Marcin.
Thanks.
Thank you.